Check test 1, 2.
Ready to go?
Yes.
Okay.
Yes, we are.
All right. If I'm going to ask them to do it, I guess I should do it too. Right.
All right. Yeah. I do the same thing.
Right. I'd like to invite those of you coming in to please come down to the front where you can see and be seen. So I'm Rich Goldberg, the director of the Cancer Institute, and it's my pleasure to welcome you to our annual Lawrence and Jean DeLynn Lectureship. So Lawrence and Jean DeLynn have been wonderful benefactors to the university. They sponsored this lectureship. I'm also the Lawrence and Jean DeLynn Professor of Oncology. And so they are believers in West Virginia University and have been generous with their beneficence. Lawrence has passed away, but Jean is, she's not with us physically here today. She's in Florida and is getting older. I did have the pleasure of meeting her last summer when she made a trip up here. But she did get a nice bouquet and note from us this morning in lieu of being here to join us. It's a pleasure for me to introduce a friend and colleague, John Bird. So as you can see, John is the D. Warren Brown Chair of Leukemia Research. He's also a distinguished university professor, an honor bestowed on only a small subset of the vast faculty at the huge Ohio State University. Laboratory-based researchers as well as a clinician, and is truly somebody that learns at the bedside and then explores in the laboratory. And for many of you who have watched the development of Ibrutinib, he did some of the initial laboratory tests on that drug, then brought it to the clinic, and really is, in my mind, perhaps not in his because he's a modest guy, single-handedly responsible for the development of this amazing drug, which is so important in the management of patients with CLL and is finding expanded uses in other parts of hematologic malignancies and even in some solid tumors. So John runs a translational laboratory. I would have to say that he has distinguished himself as an advocate for women faculty, and when he was the division chief of hematologic malignancies at OSU, he prided himself in the fact that more than half of his faculty were women, and many of the people that he's mentors and whose careers he has accelerated have come from minority groups and or are women. So at this point, he's got over 500 papers. Have you read them all? Do you remember them all? Every single one of them. And one of the things I would say distinguished him is that he would often be sending me emails at 1 in the morning, so I don't know if he ever sleeps. His main areas of interest have been CLL, but he actually has turned himself to looking at acute myeloid leukemia more recently. We were active in opening the new James Cancer Hospital, the third largest cancer hospital in the country, where there are over 100 people with leukemia in the hospital every day. So it's a huge endeavor, and John has really led it. He also continues to see patients once a week, and patients come from all over the country to see him because of his expertise in CLL. And his wife, Laura, when I was at OSU, was finishing a chaplaincy residency. So she got a degree later in life and developed an additional skill. John grew up in Arkansas. He's a University of Arkansas football fan. And we also share a passion for fishing, which we both enjoyed. So I'm really delighted to welcome John to speak to us today. I know that he will not disappoint you with his keen insights into a disease and group of diseases that he has helped to advance treatment of so importantly. So John.
Thank you, Rich, for the wonderful introduction. I'm humbled to be here to speak today. And what I would like to do over the next 50 or so minutes is tell a story about targeted therapy in 3 types of leukemia, 2 where we've seen advances, 1 where things are on the horizon, and maybe I'll get invited back to tell the story of the 3rd type of leukemia in the future where we have more success. So I'm a cartoon person. I'll just start with this. This is my personal memories of cancer care. The first is a patient in his internist's office. We found a mass. The good news is we have weapons of mass destruction. And my mother died of cancer of unknown primary in 1996, and I saw at a personal level how chemotherapy can just take the will to live out of a patient. And my goal as a hematologist and oncologist is to try to really get us away from using chemotherapy. The bad news is, even with chemotherapy, which has been around for a long time, targeted therapy has its own problems, and the second cartoon brings forth the bad news is the chemo can kill you before the cancer does. The good news is the medical bills and health insurance can kill you before the chemo. So as we've had advances, another issue that we have is our patients, and I grew up in the poorest state, the 49th poorest state, in the poorest county in that state. Thank God for Mississippi. Not West Virginia. So if patients can't get the therapy you develop, then it's a worthless therapy. And so these are sort of issues as we think of cancer care we have to consider. So I'm going to talk about three types of leukemia. I'm going to start with CML, which is a disease where we have a single genomic abnormality that led to the first targeted therapy for cancer. And then I'm going to talk about CLL, where rather than it being a genetic target, there's a biochemical dependence in the cancer cell, not the normal cells, that leads to a successful targeted therapy. And lastly, I'm going to talk about AML, where we have multiple diseases, where recognizing that rather than one disease, we have 10 diseases, and dividing those into specific diseases, and can we give precision targeted therapy to impact the disease? So let's start with CML. And CML is seen in approximately 6,000 patients. Patients have an average diagnosis of 66 years. And it was defined by a translocation of the ABL gene to the breakpoint cluster region that leads to a fusion of 2 components of a protein. So you get a oncoprotein that doesn't exist in normal cells, but that will transform any cell you put into it. So you put it into a benign cell, it will kill the mouse, it permanently transforms it. And when it's around the time that my mom was sick, CML was a disease that was treated with a drug called interferon and cytarabine chemotherapy. And if you're diagnosed with this, you had an average survival expectation of 5 years. 10% of patients would go into complete remission, though, on the interferon and the cytarabine. And you had the excitement of living your life on interferon and cytarabine, which was like living your life with the flu continuously. Not a good. Allogeneic transplant was curative, but in the 90s, we were transplanting very few people who were over the age of 60, and the median age of this diagnosis was 66. So this was a death sentence for most patients. The somebody who hopefully will win a Nobel Prize one of these days, Brian Drucker, identified a visionary person that you could target this kinase. It's present in cancer, but not in normal cells, with a drug called Imatinib. And this completely changed the landscape of CML. And this, we're looking at a survival curve here. And what you see is by decade, how the introduction of this medicine transformed this disease to where now, if we look, 86% of patients will be alive at 8 years with CML. Really the dream of targeted therapy. And in fact, this drug was introduced in 1996, and I remember we saw patients at Hopkins that were getting treated with it. And when Dr. Drucker was presenting the results, there are still patients that went on those trials, those initial phase 1 studies, that are alive today. So if you have CML today, rather than getting a transplant, getting interferon and cytarabine, you go on a pill, and you can expect 90% or more of the cases that your life is going to be as long as it would be without the CML. So what do we conclude from this story? Is CML is a disease where we have a signaling pathway that's generated by an oncoprotein that's not in normal cells, and this is relevant to all CML patients. What we also learned from this story that Dr. Drucker developed is that even though this drug transformed CML from what it was to what it is today, this drug had alternative targets, KIT. And the BCR-ABL translocation is also in Ph-positive ALL. And this translated to a variety of other cancers where this drug has been transformative, where you have this the mutation either present or an alternative target that the drug targeted. While we developed long-term side effects that some patients develop with Imatinib, not everybody tolerates Imatinib, the success of one drug brought the success of many. So we have 4 drugs approved for CML that are relevant, that will work in non-resistant disease. We have one that works in resistant disease. And this really fostered the idea that you could do targeted therapy in cancer. Everybody thought cancer was too tough, and so let's transition to CLL. So CLL is a disease, and we're going to talk about it in a little bit of a different fashion. And this is much more common. It's actually the most common adult leukemia seen in the U.S. when we look at prevalence of people that are walking around with it. And it has a natural history of patients being asymptomatic. They present often with another problem. They get a CBC. You see their lymphocyte count is up, or they have lymph nodes that their spouse or somebody points out. They go get a biopsy, and they have asymptomatic CLL. Eventually, though, most patients will progress to having symptomatic lymph nodes, anemia, thrombocytopenia. And I summarize the genesis of therapy of CLL. It started with chemotherapy from 1950s to 1991. No survival benefit, very modest effect on palating symptoms. In 1991, a drug called fludarabine, which essentially is as immunosuppressive as chemotherapy, but it had a bigger effect. We saw a minimal effect in survival, but there was some shift towards survival. But I remember the first patient that I cared for, Mr. Barrett, at Walter Reed as an intern in 1991, that got fludarabine as a second-line therapy for his CLL. Looking at his chest X-ray and seeing that the year after his CLL was gone, but he had, say, a pneumonia called pneumocystis carinii pneumonia that AIDS patients were getting at that time. And that's because this drug was taking the immune system of the CLL patient and making them like an immune system of somebody with AIDS. Chemoimmunotherapy came forward with rituximab, and that was really the first therapy that really impacted CLL survival. Again, toxicity, you can produce secondary leukemia, AML, and it didn't cure the disease. Well, really, we want to get away from chemotherapy. But CLL is a disease that doesn't have a single point mutation, a single protein that you can target. It really has a whole bunch of them, and most of them aren't targeted. Most of the genetic abnormalities are deletions. And so, in thinking about CLL and going after targeted therapy, I can tell you when I joined Mike Griever's lab and I said, I'm interested in developing targeted therapy for CLL like Brian Drucker did for CML, everybody said, that's crazy. That's not going to happen. And what I was telling the fellows and the junior faculty is that, when people tell you you can't do something, you just have to try to prove them wrong and think around the problem. And, and every once in a while, the people you're working with and yourself, say, we'll find a way to work around what's sort of standard. So while there's not a single mutation, there's something that B cells, which are the normal cells that CLL come from, are dependent upon, and that is B cell receptor signaling. And say, as time, so if you knock out the B cell receptor, the B cells die, and other cells don't have components of the B cell receptor, or they're not dependent upon it, and so maybe there's a biochemical dependence or a signaling dependence that's unique to CLL. And there are a variety of ways that you could target the signaling pathway. One is through, say, a proximal protein called SEC, PI3 kinase delta, which is in B cells, or this protein called Bruton's tyrosine kinase. Not Bruton's tyrosine kinase, but please do not get on the internet and look up BTK, because you will bring up a horrible person that's, thank God, now dead. That murdered a bunch of people in Kansas. And I'm not going to give him justice to mention his name, but that's what will come up on the internet if you search BTK. So how do we decide which to pick? And I'm not going to go through the rationale, but as we were thinking about ways to target this, if you SEC genetically in a mouse, it's embryonically lethal. So that's going to be a tough target to go after. And SIC is an important tumor suppressor gene in solid tumors. So if you inhibit it, you might promote solid tumor oncogenesis. PI3 kinase delta definitely will produce B cell, but when you knock it out genetically in a mouse, the mice will develop rectal prolapse. The food's not out there yet, so I can talk about mice. I can talk about gross things like rectal prolapse that mice get, and that's because they get ulcerative colitis, because you're taking out their T cells as well. It turns out that same thing happens in people when you give the drug. They will eventually get a disease like ulcerative colitis. So the mouse model sort of told us that there was a phenotype. Well, what about BTK? What is BTK? It's a member of a family of kinases called the TEC family. And when you look at its signaling features, it signals through the B cell receptor pathway, but it also signals through the toll receptor 9 pathway, which is important for CLL. And this leads to activation of several critical pathways that keep B cells alive. And we're coming back to those mice when it turns out that this is a little spontaneous XID mouse model where by nature, XID was mutated, and these mice have diminished B cells, absent B1 lymphocytes, and but they really have a modest phenotype in the other immune cells, and these mice live pretty well in captivity. Now, if you knock the BTK gene out completely, you see a much more profound B cell defect in the mice because you lose the chaperone and the transcriptional function of the protein. But what's important is there's a disease in people. BTK develops a truncated mutation in people. There's a disease called X-linked agammaglobulinemia, where, again, people have diminished immunoglobulin. They don't have peripheral B cells. They can get viral infections, more commonly. But if you replace their immunoglobulin, they do well. And this disease, was discovered by Colonel Bruton, who incidentally was the cafeteria at Walter Reed Army Medical Center, where I did my training. Was named after. So it's funny how things come together in life sometimes. So BTK, what's its relevance if we can't target it? Turns out, as we're thinking about this target, we're called by a company called Pharmacyclics that had a series of BTK inhibitors that were unique. They were irreversible. And Ibrutinib, or PCI 32765, was the tool molecule, not the molecule that was supposed to come in the clinic. And we got to test the molecules that were supposed to come in the molecule. And the tool molecule and this one have very potent inhibition. And they did the effect in, they block B cell receptor signaling in cell lines. They actually were given to dogs with lymphoma, spontaneous lymphoma, and they saw clinical activity, orally bioavailable. But this was a tool molecule, so it also irreversibly inhibited several other kinases, such as EGFR, ERB4, BMX, ITK, and TAC. And then it has about 30 other reversible targets. So what's the big deal about this drug being irreversible? Well, the big part is that part of this drug, nobody thought you could get, so if you went to a chemist and you said, make me an irreversible inhibitor, they would laugh you out of their office. That happened, I can attest. And that's because if the target is ubiquitous, it's important to the cell type, all cell types, you're going to see a lot of toxicity because you completely take out the function until it regenerates. There's also the possibility that you can get an adduct formation where because once the drug binds to the protein, you change the structure, it can become antigenic, and it's also it can be degraded differently depending upon how, say, the ubiquitin ligase that degrades it. However, when you have a target like BTK, it's of great importance to potentially a tumor cell, and there's a lot of redundancy in normal cell. An irreversible inhibitor is really helpful because you can inhibit the target quickly. So if you have a drug with short exposure and you don't have to dose frequently, and you can follow exactly in the tumor cell with a probe, are you seeing complete inhibition of the target by looking at the occupancy? And really, an important component about Ibrutinib is that before this, nobody had explored an irreversible inhibitor. So I'm not going to go through the preclinical data or show pictures. All I'll say is that when we submitted our paper to Blood, and the other group from MD Anderson did after us about Ibrutinib, these papers would have not gotten published in Blood. They would have gotten published in the Turkish Journal of Immunology, toenail clipping, and back scratching. And why is that? Because the data was so unimpressive. The drug didn't kill cells unless you went up to a real high concentration. And so, it really did nothing. It didn't work against cell lines. All the things that we did with chemotherapy, this didn't work. But what it did do is it stopped the cells from proliferating in a microenvironment that, like you have in people. And what the point I'll make is this sort of taught us that models that you use to test new drugs that are for cytotoxics are going to be very different than for targeted molecules, and that you have to really be open to considering other models. So moving forward to the phase 1 study and why our paper did get published in Blood, because by the time we were submitting our paper, we knew this was working. We'd actually selected the tool molecule as opposed to, I've got to stop using that term. My son would tell me to stop using that term because to younger people, tool means something else. But it was a tool. It was not intended to come in the clinic, but it ended up being the best one, and of the molecules that we work with. And initially it was given on an intermittent schedule every 4 of 6 weeks. And say anybody that says that patients can't benefit from phase 1, 11 of 16 patients had responses in the phase 1 with CLL. It was the same across a variety of B-cell malignancies. It was well tolerated. We did not get to a dose where a toxicity was identified that was dose limiting. So mild bone marrow toxicity. What we did see was early lymphocytosis, which again, this is very important because in CLL before these studies, when you see lymphocytosis and you give a therapy, it's a sign of progression. And, so there was a lot of uncomfortableness with this and whether this was progression. And actually, some doctors at other places talk to the patients, well, you're progressing, you need to come off therapy. And the patients get credit for helping move this forward because they said, I'm feeling great. My nodes are smaller. I'm not stopping this. And the lymphocytosis resolved over time. The major toxicity that was seen with this was rash, heartburn. There was some early AFib, atrial fibrillation in a small number of patients, and loose stools. And again, most patients are older with this disease, and most of them did not think of the loose stools as an adverse event. So we flash forward to the phase 1, the phase 2, and this was a pivotal study. This study led to the approval of the drug. Patients with untreated, we were seeing activity in previously treated patients. So there was a small pilot group of patients over the age of 65 who were symptomatic, who didn't want to receive chemotherapy or were not candidates for chemotherapy. And then patients who had received a bunch of therapies. And the median was 4 prior therapies, but there was one patient on this therapy, my own, that had 14 prior therapies for their CLL. Most were high risk. And again, we saw the lymphocytosis, but it was ignored. And, I'll just take a diversion here and say, if you think developing a drug for disease is tough, that's easy. The hardest thing of the development of Ibrutinib was getting the CLL experts to agree to a new response criteria that disregarded the lymphocytosis. So that was say, and getting everybody to agree that these were beneficial findings. So we flash forward 7 years of follow-up. This looks at the progression-free survival, and there are very few patients with untreated disease that receive this drug as initial therapy. Even in the relapse group, over 40% of patients, at 7 years are still progression-free. And again, these are older patients. The patients on this lower dot who had a lot of therapy, the median life expectancy for that group was probably 6 months when they went on study. So big difference. The overall survival is shown here, and again, we're seeing the same. We're seeing the same thing. Just very, very flat until the curves go out. What about side effects? This drug does have side effects. Most of them are acute atrial fibrillation. The things we talked about. We do see over time hypertension increasing, and there's a small cardiovascular with this that we continue to try to identify who this is going to happen to. When we go to the phase 3 study, and that really proved the validation of this. And there's the first one was the RESONATE-2, which compared chlorambucil to Ibrutinib, which had never been done. Horrible study. Chlorambucil is not an appropriate control for CLL, but they showed that it improved response, progression-free survival, and overall survival. But it was really these 3 bottom studies that I outlined that looked either in elderly patients at bendamustine, rituximab versus rituximab Ibrutinib versus Ibrutinib alone that showed a significant benefit to Ibrutinib over chemoimmunotherapy, and then a younger study done by ECOG that showed FCR was inferior to Ibrutinib and rituximab. And then the company did their own study looking at chlorambucil and obinutuzumab. All the same story. Ibrutinib is a much better drug than what we're doing. So this is great, but what about Ibrutinib's moved to the upfront setting. What about patients who have to discontinue because of intolerance? And I say 10 to 20%, but really in the real world, this might be as high as 40 to 50%, depending upon the age of the patient. And this can occur for a variety of reasons that are shown here. I've left out financial, but this is an expensive drug, and sometimes patients won't take this because of the cost. And so what to do here? Hopefully we've made the case that BTK is a great target and produces durable remission. So if people are intolerant, what are we going to do? And so by virtue of being involved in developing Ibrutinib, we were able to look at a lot of second-generation medicines. And there's this one called Acalabrutinib, and this is a kinome scan, and you see Ibrutinib's hitting a lot of different things. Acalabrutinib is much more selective, and it doesn't hit some of the kinases that cause adverse events, such as EGFR and ERB4. And so Acalabrutinib came forward, and again, in a relapse patient group, 134 patients, say with relapse and refractory disease, again, most refractory, and we saw a much better safety profile, no atrial fibrillation, minimal myalgias, paniculitis, other things that can occur with ibrutinib. And this just shows the side effects. And what we saw was very similar. The time to response was shorter. And when we look at how this drug is comparing to patients with on ibrutinib, and again, these are two long-term follow-up studies. The longest one, obviously, with ibrutinib is the first drug versus acalabrutinib. They look the same in the untreated setting, and the demographics of these patients are the same. Obviously, they're going to be randomized studies, but even if they're the same, the bonus is you have less intolerance. You don't see atrial fibrillation, hypertension developing over time, the myalgias, arthralgias, or GI symptoms. And so, so what about, though, for the patients who are intolerant? Because that study doesn't really address that. And actually, as part of this phase 2, we enrolled 33 patients who had to go off ibrutinib because of intolerance. And what was interesting is 64% of those patients that were treated with acalabrutinib started that and had no recurrence of the adverse event that they'd had with ibrutinib. The remainder, with the exception of 2, had diminishment of this. We did see in people that were coming on and off ibrutinib that there was a higher frequency of development of the mutation that's associated with resistance, which we're studying right now to see if it pre-existed. So, what about the phase 3 studies? Well, it turns out there are 3 phase 3 studies that are being done with this. Two being done just like with ibrutinib, that looked at either younger patients or older patients, and in relapse disease, and all have been positive, just like ibrutinib. And there actually is a phase 3 study comparing both of these agents in relapse and refractory CLL that's accrued. We're waiting on the outcome. So that doesn't get to the issue, though. We talk about intolerance. What about the patients where it stops working? And this looks at the bar graph of our first 308 patients treated. And again, with a fall at this time, this should be 4.3 years. We see that a good number are still on therapy. But other patients went on the transplant. We had a subset, though, that progressed. And we got to say, when you look at the frequency of patients progressing on ibrutinib, you see that those that transformation to a more aggressive lymphoma, risk of CLL progression continues to go up over time in relapse patients. And then the other events continue to go up in time. And the outcome of patients for these different groups is very different. So, and here we show patients who go off who don't have an infection. And this is likely reflective of patients having other options at the time of this. This is a CLL progression. Again, most of the patients still are passing with time. And this is Richter's transformation, where we're still we have a lot of work to do. So, what do we know about ibrutinib resistance? So it turns out that the drug irreversibly binds at the to the cysteine residue at the 41 site. And by studying patient samples, we were able to identify that about 75% of patients will mutate that point site where the drug binds and make it go from an irreversible inhibitor to reversible, a very poor reversible inhibitor. Another 10% of patients will develop a mutation right downstream of PLC gamma 2. This discovery came from, so while I'll show the cumulative data of a lot of patients, this came from 5 patients who we studied samples for, 6 patients we studied, and 5 had one mutation, 1 had the other type of mutation. So diagnosis to relapse samples and having access to those and studying those, you can learn a lot. For Richter's, we still have no idea what the pathophysiology of transformation is. It's probably an epigenetic mechanism. It's understudied. Looking at our experience, as I said, 87% of patients will develop mutations either where the drug binds or to a kinase immediately downstream. And sometimes patients will have both mutations. One of the questions we wanted to ask, because if any of those of you who are hematologists that have treated patients that have gone on Ibrutinib, when they truly become progressive on that therapy, their disease will explode. And we saw this in a few of our early patients. And we wanted to develop a strategy to, if we could, identify it. And this just shows in samples that we collected over time. And when we saw this was working, we started collecting samples from all of our patients. And what you see is, if you work back, this is where they relapse. And you see if you work back, each of these red spots are where you could detect the mutation with a sensitive assay. And on average, you could, from our retrospective samples, you could detect the mutation 9 to 12 months before the patients would relapse and become sick with the disease. This can be done we started doing this prospectively because we didn't really want to be interfering or to be managing patients when they were on the brink of death with progressive disease. And this is sort of something that we now do, monitoring our routine patients with this assay. The question, well, does the mutation go away when you stop Ibrutinib? It doesn't. It actually will increase with most therapies. Really, the only thing we've identified are some immunotherapies and Venetoclax that will get rid of it. And so, what are some strategies to treat resistance? Well, one of the therapies that I'm most excited about is developing a reversible BTK inhibitor that's not dependent upon that binding site or something that degrades the protein, because the CLL cells are still very dependent upon that. And we'll talk a little bit about that. Some other B-cell receptor signaling agents, immune therapies such as TILs, which we're very excited about, Venetoclax, which I showed clears the clone, and I alluded to. So there are a variety of things that we're working with in clinical trials. Targeting BTK with a reversible inhibitor is something that there are two promising drugs, and one that the company sort of walked away from, GDC 0853, but the two that are promising during clinical trials, ERQ531 is a more broader inhibitor. It has a half-life that's going to allow complete coverage of the mutated kinase, and then LOXO305, which is a more selective reversible inhibitor that just went into the clinic. And this shows sort of the biochemical features of RQ. It's broader than Ibrutinib, as you can see here. Has a long half-life in multiple species. If you get the micromolar concentration, and Jennifer Wayak, one of my former fellows and the CLL lead at OSU, published this in Cancer Discovery. Part of why I'm excited about this drug is we like to work with approved drugs are going to work or not work in mouse models of CLL, and this medicine is one of the few drugs that we've given that's a BTK inhibitor that actually beats Ibrutinib in our mouse model. It also works in a Richter's model of CLL, and this just makes the point. This is a gel showing that the drug in a patient sample prior to relapse, we're seeing Ibrutinib is inhibiting BTK. RQ is as well. And then when we take a sample from a patient at relapse and we stimulate B-cell receptor signaling, we see that RQ inhibits BTK, but Ibrutinib doesn't because it can't bind to the drug. That's a Western blot in a mouse experiment. Let's get to where it really matters. This is a patient with Richter's transformation, and we see this area, and after 3 cycles of therapy on this patient's had a dramatic response. This is not a cure for Richter's, but we've seen this in several patients, and this enables us, even with Richter's now, to have an option for some patients that we can get them on to CAR T cells or something else. There are a variety of alternative strategies for resistance. Ideally, we want to prevent it. So sequencing our therapies to optimize the immune system, say, and then also something that we're doing is, say, and really this is a paradigm that Ibrutinib and other irreversible kinase inhibitors allow us to do, is when you can identify the main cause of resistance, and it's a lot easier to treat 1% of the cells that are developing resistance by adding something than waiting till the patient is sick and you have a fully resistant clone. And so this is a strategy that we're taking on with adding on to molecular relapse. And then ideally for Richter's transformation, say targeting it with specific agents. And we're interested in bromodomain inhibitors with Ibrutinib and also the ARQ in high-risk patients. So how can we prevent acquired resistance from the beginning? And a lot of people are saying, well, let's combine all the targeted therapies. And I've not talked about Venetoclax. This is another say agent that targets BCL2, and our group and several other groups have combined it or Acalabrutinib with Venetoclax or by itself, and you see really high responses, and including about a third, in some series, half of patients getting MRD negative CRs. And looking at sequences, is another thing to do. And really, the whole field of CLL is just emphatically excited with combining Venetoclax and Ibrutinib or Venetoclax and other BTK inhibitors. I am too, but it's a clinical trial, and we still don't know that we still don't really know all the long-term effects of Venetoclax. We've got to follow up on Ibrutinib. And I show this because the ones I'm asked, well, what do I think about giving Venetoclax and Ibrutinib together off clinical studies? And I don't do it. And I fought hard when we talked about this in the NCCN guidelines, looking at the immune cells when, so you see no change generally in T cells or NK cells when you give Ibrutinib or Ibrutinib with an antibody, but look at the NK cells. And this is 3 months after stopping therapy with a triple therapy. This is 8 months after. No NK cells, very few NK cells. And same thing with T cells. And so, and the one phase 3 study with Venetoclax and Obinutuzumab is the only phase 3 study that's been done in CLL where you actually, you saw an improvement in progression-free survival, but there were more deaths of patients in the experimental arm. It wasn't significant. And so we have to have a pause until we have the data before just jumping on the bandwagon of combination. So for CLL, I think we can conclude that B cell receptor signaling is a biochemical pathway that can be targeted in all CLL patients. If somebody calls me and they say, I have a CLL patient, I gave Ibrutinib to them, they didn't respond, my first question is, did the patient have CLL? Because I've only seen 1 or 2 people that I'm convinced that didn't respond to Ibrutinib, that had CLL, and they were usually very weird cases. The impact of targeting has been very significant for CLL patients. Older patients will live as long as they would without CLL if they receive this class of drugs, inferring from survival curves of patients without CLL. The second generation molecules are exciting because they offer potential improvement, but also better drug tolerance. Resistant monitoring may allow a new treatment paradigm for this disease. And finally, we'll see if the combination therapies are better, or if we see more toxicity, that as patients are living maybe as long as they would without it, that we need to to save therapy for sequencing. In the last 45 minutes, that's a joke, I'd like to just talk about AML a little bit. And so when we think of targeted therapy, AML is really multiple diseases, and I'll hopefully make the case of that. And maybe here, a precision medicine umbrella trial for moving forward with targeted therapy might change the outcome of this disease. And so, when we think of precision medicine, what we don't want, the patients with the nurse, somehow your sequence result got faxed to a complete stranger. He has no idea what's wrong with you either. But really, here's what we want: the patient having their sequence for their disease and going to the pharmacy and getting a medicine, ideally that they can afford. So as we're thinking of precision medicine studies, there are 2 types. Say in general terms, An umbrella study will give different drugs that treat different mutations in a single type of cancer. And a basket study is where you go after one pathway or one mutation and you bring in different types of cancers that have that. And so as we think of AML as the disease, it actually is the most common, but not the most prevalent type of AML. It occurs in elderly. And up until 2 years ago, there have been no therapies approved for AML, say for more than 3 decades. We were treating AML at OSU just like Rich did when he was an intern 60 years ago. Oh, sorry. So for simple terms, you can think of AML as 2 diseases. One where you have balanced translocations, and that typically occurs more in the younger patients. And then mutation-driven AML that occurs much more common in the old, where you just get successive mutations that ultimately lead to development of a fatal disease. If you look at the sequence of patients with relapse, though, one patient often has 10 diseases in them. So if you're going to really do targeted therapy, you have to, with one drug or one set of drugs, you need to do this early. And this just goes through the pathogenesis of AML, where you start with one mutation, several, 3, and then 5, you have the disease. And we really want to go at targeting here before the patient develops AML and get to the trunk of the disease. And we have a variety of molecular targets in AML that we didn't have. And so this whole idea, can we do a trial in AML similar to what's been done in solid tumors, where we have a multi-arm master study with each arm independent of one another, so you can test multiple drugs. We also wanted to do a study where we could bring junior and senior AML clinical investigators to lead the trials and teach the next generation how to do small clinical trials. This study is designed to look at for large effects. So, and hopefully there's a statistician that I'm not going to offend in the audience by saying this, but if we need a statistician to tell there's a difference in the arms of our study, then we're off target. We're looking for big differences. We want to do small studies that can ultimately lead to accelerated approval. And the why of this study is to bring safe drugs that are effective to patients faster. So the trial is designed like this. Say patients come in, they're untreated, they give a sample, and we send it off to 2 labs. And say genomic sequencing is done, we stabilize the patient for this 7 days, and then the results come back, and then we assign treatment. And this is done centrally by one of the principals. Our cytogenetics are done centrally. And again, during this time period, they're stabilized. And this is a big deal, because typically you treat AML, say, the same day or the next day. You don't let the sun set without treating AML. And so could this be done safely, and was it feasible? That was a question. And so how do we assign therapy? Based upon picking the dominant clone and also the clone that has the chance of curing a patient. And there are 2 up here, and then we sort of go down with a variant allele frequency, or the proportion of cells that have a mutation of 0.3, and then we do 0.2. And for time reason, I'm going to say that we validated this method with a big set of patients from the alliance that received 7 and 3, and it worked. That we saw that with the majority of patients, say we could assign based upon the dominant clone. We only had to go to 0.2 variant allele frequency in 10 patients. Let's get through this. So how does the study work? The Leukemia Lymphoma Society, well, that's not a pharmaceutical company, that's a philanthropic organization, holds the IND for the study, but we run it like a company. So industry comes to us, say they want to put their drug in our trial. We have a CRO that monitors. We use a lot of novel technologies, such as Protocol First, that allows us to bring the cost of our trial down to about 40% of what, or 60% of what an industry would typically pay for the study. We have our own independent data safety monitoring board. And again, this is a team effort. These are our sites. So right now we have 16 sites. We started with 4. And say from November of 2016 to this is our data cut May 10th of 2019, we enrolled 588 patients. These are patients over the age of 60. People said this couldn't be done. 478 had AML. More than half went on to one of our sub-studies. 219 decided to get standard therapy. Some of these went on to alternative trials that were better at the site. I call attention, 44 patients did palliative care. Significance of that, these are older patients. They had the time over 7 days to think about, do I want to take therapy or not, and make a decision, versus being pushed into going on to therapy that they ultimately decided they didn't want to do. So this, to me, this is an impactful thing for allowing people to decide what they want to do versus moving them into therapy before they understand. How successful was it? We were able to assign therapy in 95.5% of patients within the first 7 days. The trials we have open are shown here. We have a variety of targeted agents where we either give a targeted agent plus a standard therapy, or if we have a real active drug like Imatinib, positive disease, we'll give a monotherapy. The results of the study, we had 2 studies last year that were presented at ASH that they continue to be very positive. We've had 3 meetings with the FDA this year, and we're hoping to bring 2 supplemental INDs forward for 2 of our genetic groups. Where are we going with this study? Well, what's fun about drug development is the field. There's this drug called Venetoclax. Has anybody heard of that? Yeah, so if you're an AML doctor, that's for some patients, this has been transforming. So we had to adapt our trial to a new standard of care coming forward, very similar to what the MATCH study had to do in lung cancer. We've learned that there are certain molecular subgroups, such as those that have RAS mutations, where most of our therapies, even our targeted therapies or epigenetic therapies, don't work. So we're developing a RAS group specific. So we're adapting quickly. We're really moving to novel combinations where, based upon the laboratory-based hypothesis, we're extending to younger patients. And the conclusion that we've come to is, in the data we presented at ASH, we are shifting the curve of patients by doing this for survival. But we're not curing enough patients. And the best way to treat AML, we believe, is going to be to prevent it. And so we're starting a second study in myeloid dysplasia, where we're going to go after things earlier. So what's the most important part of this study? How did we go from starting this 2 years before in November of 2014, November 2016, and now having, as of yesterday, 702 patients on study? Our principal bylaw, we have a bylaw for this group, for this study, and that we agreed on. To be involved, you cannot care about credit unless you're a junior investigator at the time of study completion, because you need a publication for promotion. And if you care about credit in this study, you're out. We've thrown a site out that said, we're not getting enough credit for what we're doing. We're going to do our own thing. I'm the CMO because we had somebody from industry that came to the Leukemia Lymphoma Society and said, I'm not getting enough recognition for what I'm doing. So this is a core thing. And I think Rich will agree from his work with colon cancer, Clay, with that if you're successful and not just move forward, if you're successful in doing something, at the end there'll always be enough credit to go around for everybody. I personally learned that with the story that I told with Ibrutinib. This is happening with Beat AML, and so much progress gets hindered because of this concern. So hopefully I've made the case that disease-specific genetic or biochemical targeted therapies can greatly change the history of different blood cancers. Now, biochemical pathways can be targeted with an irreversible inhibitor. You'll see a better result in general, although we will test this hypothesis. Targeted therapies also offer the opportunity to change the paradigm for treating cancer if we understand the pattern of resistance. And this is sort of something that we're actively working on. And finally, precision medicine offers the promise to change the treatment approach to AML. We're really excited about the direction this trial and effort is going. And there'll be a lot more data about this study at ASH this year. So I want to acknowledge for the Ibrutinib work, our own CLL team at OSU. Amy Johnson, who did all the preclinical work, Jennifer Wayak, who heads our CLL group, our computational biologist, Jane Blatchley, students and technicians, clinicians, those at other sites that were actively involved, say in this story. Again, it was a team effort. And say our society investigators, principals of the study, and finally those people who support us and invest in the work that we're doing to try to change the natural history of blood cancers. Thank you. And yes, it was 60 years ago.
So John, that was quite a tour de force. I had two questions, but I could ask about 100. So when you look at people that respond initially and then stop responding, how frequently are these epigenetic problems versus how frequently are these other sort of mutations and other critical genes?
With the irreversible inhibitors, 86% of patients have point mutations. Say with when they relapse with the de novo disease, if it transforms to something else, it's almost we believe it's almost always epigenetic. With reversible inhibitors, there's often and with epigenetic inhibitors, it's almost always a signaling shift or an epigenetic shift. So it's different by the class of drugs.
I was wondering though if you would pair epigenetic inhibitors, which did in the last one, say in CLL, when you talk about Richter's transformation or some other things with the irreversible inhibitors you're using.
Gosh, this is the topic of a grant that we have in review right now. So we are, thank you. But we are probably the bromodomain inhibitors, which get after epigenetics. We're real excited about in Richter's, because in our preclinical models, I didn't show the data, they cure the disease.
That's cool. The other question I had is anybody trying to mess up the microenvironment at the sort of the more cellular or the more infrastructure part of the microenvironment as opposed to a cell basis, like looking at fibrin or other kind of molecules to disrupt that?
That's being done in AML. And there've been some early successful results. A challenge that we're having in CLL right now is we still have a lot of way to go, but unfortunately there are diseases that are a lot where people are doing a lot worse, and so the priority of actually getting funding to work in CLL right now is hard. So the principle. But that principle could potentially work in CLL, but it's going to be hard to show the difference because the current therapies are doing so well.
Excellent presentation. I have a lot of questions like Dr. Clay, but I have to restrict a couple. First of all, what about your opinion about curing CML? At least Europeans are claiming that if a patient is really early detected and treated, and after 6 years they can withdraw treatment and they will be completely cured. Second, Dr. Drucker in Oregon has this IP 1371 Gleevec, but there is another history often is not told, the structure of the able kinase pocket. Crystallography has discovered or published 2 years before by MIT, a scientist called Chaco. And based upon that only, Novartis synthesized the ATP structure binding and devised this particular inhibitor. And it was in the freezer for a long time. And that is taken by Drucker. And then it was given and it is effective drug. That is not told by how basic science, very fundamental science, by crystallography, aided discovery of this competitive inhibitor. And also, really personal medicine, you are thinking about that, you have to think about ultra, PML, AML, you have complete cure. So AML is not completely not curable. If a 15-17 translocation, if easily detected early, and if patient is taken, arsenic trioxide and ultra, arsenic trioxide 100 years old, can be cured, that terrible diseases.
No, so I'll address your first question. and I'll make a comment on your second question. So your first question, if we introduce Ibrutinib or targeted therapy early in CLL, will we change the natural history? And there are studies that are ongoing. I think that, and I think there might be a subset of patients where where that happens, where you can reestablish the immune system, but the studies are ongoing. Your second comment is, I singled out Brian Drucker because he's my hero, and and there are a lot of people that deserve credit for, I mean, he was at Harvard before, he was at Harvard for a long time, for a long time before, and before going to Oregon. And I think there were a lot of people involved in the success of of that drug. And but I think we would both agree, that he was one of the visionaries that took the basic science and moved it to people when nobody else would. And I don't believe that we would have Gleevec or the targeted therapy for CML if he and Charles Sawyer and some of the other people hadn't really pushed it. You can disagree.
Thanks, sir. One more question. You have to understand, BTK inhibitor also 30% or 40% of 17p deletion to respond. But we know that those CLL patients who have 17p deletion or ATM deletion, 50% of them will go to rectal transformation. On your Ibrutinib and other studies, you have taken into that consideration and rectal transformation when you are thinking about genetic background of the patient?
Yeah, well, I think 17p patients who get Ibrutinib as first-line therapy, the frequency of relapse, even with extended follow-up, is very small. There probably is something else. Is when you're giving a 17p patient that's had 9 therapies, there are other things that are driving relapse. So I think as you move these drugs early in the course of the disease, resistance is going to be a rare problem. I've seen one resistant patient to upfront Ibrutinib in the entire time, in this entire story of my practice.
Does ARQ-531, when you're especially giving it in the relapse after Ibrutinib, alter the T regulatory cells in the tumor microenvironment or not?
That's a great question. We've not looked at it. We've not looked at that carefully yet. And I would guess it might. As I say, it's much broader. But we certainly have seen robust responses in that patient group.
Especially knowing the tumor microenvironment will be more immunosuppressed after the use of Ibrutinib.
Yeah, absolutely. So when you get this mutation, when you get the Cis41S mutation, is unlike most mutations that develop or have less activity. This one has more activity. So if you put that mutation, if you put the BTK with this mutation into an XID cell line, it will kill them. It will engraft faster. It's more immunosuppressive than the native protein. It has more native intrinsic kinase activity.
Thank you.
Yes.
Thanks for your incredible work and sharing it with us today.
Thank you for having me.
I was interested in, if you had another Ibrutinib today, and you kind of were in the same situation where you didn't have an MTD and you're trying to select a phase 2 dose, would the decision, the type of data you were using now be different than what you used then? And what did you use then to decide what to go on?
What was used in the phase 1 was they went two doses above the complete occupancy. And if I had a good occupancy assay that I could test in the tumor cell where I showed that we had 99% or more occupancy, then I would stop unless there was a compartmental difference. And some drugs, there may be a compartmental difference between the node and the blood.
One last question here.
Great talk, thank you.
Thank you.
You talked about preventing AML with regards to treating MDS. Now my question is with regards to some of the conflicting data about clonal hematopoiesis, one, and number two, Also, how do you envision selecting those patients, like risk stratifying them, and how do you envision selecting those patients to go on these targeted therapy?
Yeah, it'll be, so the study that we will initially do intermediate high risk and, say identify mutations where you have obvious drivers, driver mutations. We still don't have good spliceosome inhibitor drugs, for instance, and for that group of patients, we're going to rely on synthetic lethality studies. Like, so one of our first drugs will be an ATR inhibitor, for instance, which has synthetic lethality with spliceosome. And to your question about clonal hematopoiesis, that's an incredible, exciting area because it brings toward cardiology, toward other developing solid tumor cancers, and but there are a lot of people that have that are going to die of something else, say may or may not be related to clonal hematopoiesis. So I think you're going to have to go with really, very gentle drugs if you're going after that, and it's going to be big studies, and say probably they'll be completing by the time my grandchildren are retiring. So thank you.
So I want to thank John for spending the day with us. We didn't just have him give a lecture, but he met with many people today in a way that I hope was beneficial to everyone that had a chance to interact with him. You could argue that what he's done is somewhat heroic in its own right, changing the treatment paradigm for CLL and really changing the treatment paradigm for using kinase inhibitors. So this is really important work that I don't want you to underestimate the importance of as you're listening to what he said. We do have refreshments outside. Part of the endowment for the DeLynn Lecture is having dinner with President Gee, so you'll have to excuse us when. Yes. So please join us outside in the lobby for some refreshments, and apologies in advance that we're going to have to leave in about 10 minutes.